Alright, the last section in Chapter 8 that we'll look at is Section 5, 8.5, that looks at different types of mutations. So the previous sections have looked at DNA, how the DNA gets transcribed to RNA to be translated to make proteins for all of the different processes within the cells, whether they are our cells or bacteria cells. However, within DNA, there can be mutations can occur, and then that mutation within the DNA sequence can then be carried over to the mRNA sequence, which can then be carried over to the protein. And so you can have mutated proteins that then don't function properly, and in some cases do something completely different, and those can be harmful for our cells and cells around us. So what causes mutations? All right, some mutations can actually be spontaneous, meaning there's just some random change in the DNA, and that random change is kept within the DNA sequence, and it just allows for errors downstream of that. So within the DNA, after it's been replicated, duplicated, then in the mRNA sequence, and then potentially in the protein sequence. There are also what are called induced mutations. And induced mutations, something causes it to happen. So if you think about inducing labor for a pregnant woman, right, that woman is receiving hormones to induce her body to want to give birth. All right, so mutations can be induced, and generally there's going to be some sort of exposure to a mutagen, something that causes a mutation. And these are generally going to be physical or chemical agents that alter the DNA. A big one is going to be radiation, whether that is UV light, and that UV light can be from the sun, which is why you actually don't want to be exposed to UV lights a lot, wearing sunscreen during the summer to block the ability of the UV rays coming from the sun to penetrate your cells and to get into your cells and cause mutations. And then chemicals. All right, here there's nitric acid. There are a lot of chemicals out there that can cause disruption to DNA. All right, so those are how mutations can occur, spontaneous or induced. And then there's different categories of mutation. So one type of mutation is a point mutation. And a point mutation is just one nucleotide, just a single point within the whole strand of DNA that is mutated. And so that can be due to an addition or deletion of one base pair. All right, so if the sequence was ATG and you added another A after the first A, that point mutation would lead to the DNA reading AATG. All right, then if it's deleted, maybe you pull out that T, and then your sequence would read AG instead of ATG. All right, then you can substitute. So maybe instead of ATG, when the sequence was being replicated, instead of adding a thymine, that T, the machinery accidentally added an extra adenine. So then the sequence read AAG. All right, then there are missense mutations. And missense mutations is any change in the code that leads to a placement of a different amino acid. So it changes, it doesn't make sense anymore after you have that mutation. And so what happens is, since you are changing the amino acid sequence, all right, because you're changing an amino acid, all right, now that can cause a faulty or non-functional protein. And this is going to be because the amino acid sequence, the amino acids have different properties. And those different properties yield to the ability of a protein to fold on itself to be able to pick up or hold the proper shape. And if you remember about proteins, shape is very important to function. And if that protein may not work at all, could be non-functional, or it can produce a protein that functions differently. Let's say an enzyme. If the enzyme's active site is different because the shape is different, because the amino acid sequence is different, then it's going to act on a different substrate. And that lock and key fit isn't going to work properly, but maybe there's a different key that can fit into that lock, so to speak. And then another component, or another outcome of a missense mutation, is that even if a different amino acid is added to the protein sequence, it may not actually affect the protein at all. And so it can also cause no significant alteration. So it's kind of a luck of the draw, really. If you have a mutation, does it do anything? Does it not do anything? All right, a nonsense mutation changes a normal mutation into a stop codon. And what that does is then it causes the protein, the process of developing a protein, so translation, to stop. And so if your amino acid sequence should have read for a protein to be 100 amino acids long, maybe that nonsense mutation caused a stop codon to occur, say at amino acid 50. And so now your amino acid sequence is only 50 amino acids instead of 100, and that's probably going to affect the protein. It's going to be called a truncated protein or a truncated sequence because it's not as long as it should be. Right, there are silent mutations. All right, a silent mutation alters a base. All right, that's going to be within the DNA sequence, but it doesn't actually change the amino acid sequence. And that can occur because there are multiple 3 base codon sequences that can code for a single amino acid. And so you may not alter what amino acid occurs with a base change. And if the mutation is silent, then it doesn't have any effect. You can have what's called a back mutation, when a gene that has undergone a mutation reverses back to its original base composition. And this probably doesn't occur very often, but it can occur. Frame shift mutation, that's going to occur when one or more bases within the DNA is inserted or added, or deleted or taken out. And so what occurs then is it changes the entire reading frame of the mRNA sequence. And then nearly always, this is going to result in a completely non-functional protein. All right, table 8.8 is a good example of these different types of mutations. All right, so in A, you have the DNA sequence up top, and then the RNA sequence, and then the protein sequence. So AUG within the RNA sequence always codes for methionine. ACC in the mRNA sequence is going to code for a threonine, and so forth. And this is the normal gene. All right, with B, you have what kind of mutation? All right, a missense mutation. It's just a substitution. So instead of this section of DNA reading CTG, like it should appear, it reads CTT. The RNA sequence is then GAA, and you have a different amino acid here than the amino acid in the normal gene. All right, so this leads to an amino acid switch, may or may not function well. It's just hard to say. In this case, you have a base substitution. And instead of CTG, G, like you have up here, the normal gene, you have CTA, and that is still going to code for the same amino acid. So this mutation is silent because there's no change in function, no change in the amino acid sequence. Now, here, in this sequence, you're pulling out this guanine. All right, so instead of being TGG, CTG, you have what is now TCC, that G gets taken out. And so when the amino acids are being added, it still is adding a threonine like it does up here in the normal spot. But everything from here on out is altered. And so now you have a threonine being added instead of an aspartame, and then an arginine, and then a stop codon. So it's a frame shift. It shifts the frame of the amino acids. And this frame shift results in a premature stop. And now your protein is going to be a truncated protein. All right, here you also have a frame shift, but this frame shift is due to an addition of a guanine in the DNA sequence. So the methionine threonine is correct, except now instead of the aspartame, you have an arginine. And it's going to alter or shift the remaining sequences and how the sequence is going to be read for the amino acids to be added to the protein. All right, so positive and negative effects of these mutations. The mutations are generally permanent. They can be passed down onto new cells, and this can occur in all types of organisms, whether it's humans, whether it is bacteria, viruses. Generally, spontaneous mutations are not going to be beneficial. However, a small number of these mutations can create variant strains, and these strains can be more readily adapt and survive and reproduce in certain environments. Is that good or bad? All right, that could be both. It could be negative to us if a pathogenic organism figures out how to survive better in our bodies, say in the gut where there's acid, or in the stomach where there's acid, or when the pH is not normal, say in the urine. All right, so those would be negative to us humans, but would actually be positive for the bacterium. Now, as long as the environment is stable, mutants only comprise a small percentage of the population. All right, so in most cases, the population is normal or has that normal genetic material. When the environment changes, some mutants will be equipped to survive in that new environment, and so the whole population will shift to this slightly different population. And how this occurs, I mean, one big area in which this occurs is going to be drug resistance, and bacteria being able to acquire drug resistance. And then they're able to go through this process of selection and adaptation within the body when a person is on antibiotics, right? Those bacteria no longer die when they are exposed to those antibiotics. And then instead of having normal susceptible bacteria in your body, those normal susceptible bacteria are killed off by the antibiotic. But then those few mutants that are able to survive the antibiotics will grow up and flourish, and then that is the main new population within your body. All right, so concept check. Which of the following types of mutations will have the most devastating effect on a cell? Cue the Jeopardy music, and the correct answer is going to be B, the frame shift. Because remember, if you add any sort of mutation in that DNA sequence, it shifts everything from that point on, and it can very easily alter the amino acid sequence and sometimes even add a stop codon. And that is the end of section